Hi, everybody. This is Joanne, Science Goddess on Twitter, and I am joined here by my co-host Jeff Schomeyer on Read Science, where we talk to popular science book authors about things they've written about science and scientists, and to give us a whole new perspective. We also like to talk about the importance of communicating science to the general public. Our guest today is a returning guest, David Quammen. It's been a few years because we last talked to you when you wrote Spillover. And
right.
Yeah, welcome. We're glad you're here.
Thank you. It's very nice to be back, Joanne and Jeff. Good to join you.
So for those of you who, I don't know, have not heard of David Quammen, I would say shame, but it's okay. We don't want anybody to feel bad when they come to Read Science. So, but basically, David's an award-winning science, nature, and travel writer, and your work has appeared at National Geographic, Rolling Stone, New York Times Book Review, and you are in the library, Study Room 4 in Bozeman, Montana. This is so exciting.
That's right. I'm at the public library. God bless the public library here, because my home office is a little bit inhospitable to focus talk right now because there's a roofing crew tearing shingles off of the roof and hammering new ones onto it. So my wife and our two borzois and our cat and our ball python are there absorbing this on my behalf, and I'm here at a quieter place.
Nice. You have a python? Oh my gosh.
We have a python. Yeah, we have a rescue.
Okay.
Back in May, my wife came down the steps one day, my wife Betsy, and said, "Don't get mad at me, but I've agreed to adopt a python."
Wow.
And I said I wasn't all that surprised, because it's the kind of thing that she does. And I am a snake person. I love snakes, and I've had a lot of snake guilt because I had pet snakes when I was young, and I didn't know how to take care of them, and they didn't get good care. So now we have this one. He's 4 foot 2 inches long. He's a ball python. His name is Boots, and he has a Cadillac healthcare plan, and he lives in my office.
Wow. Wow, this is fascinating, and wow, we might get to your book too. I didn't, I don't know, how much do I know if you're a snake guy or not, but that's pretty cool. So
if we were in my office, Boots might be crawling across my keyboard right now.
Oh, really? Really?
Yeah,
okay.
Yeah. He crawls around the office while I'm working.
Wow, and you don't have any fear of being just wrapped up by him on your leg or something?
He's very sweet and very gentle. Yeah, a little bit shy. Sometimes if you make a sudden movement, he twitches and jerks away, but it's just him being nervous and no need for you to be nervous.
Mm-hmm. Okay, interesting. Wow. Well, now new fun fact about David Quammen that I didn't know. Yeah. Anyway, so today we're going to talk about your newest book called The Tangled Tree, A Radical New History of Life. And I've known about this book for a little while because you had to come to the University of Illinois in Urbana-Champaign to interview many people for this book, including people who knew Carl Woese.
Right.
This was
Carl Woese. Yes, Carl Woese is the central character of this book. It's not a biography of him. It's a book about a constellation of ideas and facts that are enormously counterintuitive and surprising and significant for our understanding of the history of life on Earth. But at the center of this constellation of facts and ideas sits this one guy, one microbiologist who worked for 40 years at the University of Illinois, there where you are, Joanne, in Urbana-Champaign. And he triggered a revolution in thinking that began this discovery of what I call in my subtitle, my modest subtitle, a radical new history of life. And that's not my radical new history. It's a radical new history that comes from Carl Woese and the people who were influenced by him. So yes, I came to Urbana a number of times to talk to people who knew Carl Woese, who worked with Carl Woese, to spend time in the University of Illinois archives going through the Carl Woese papers. He is the spider at the middle of this beautiful big net that I've tried to capture in the book.
That's great. Hey, Jeff, I think I sort of took your first question.
That's okay, because it is central there, and I noticed that David snuck in the word web, which will become burdened with significance, I think.
Right, and I think I said a spider at the middle of the net, but what I meant, of course, Jeff, was a spider at the middle of the web.
There you go. Which we'll get to. And I was fascinated by the title of the book because I got some of the allusions, Darwinian and others, to that idea, but I had to read more than half of the book, I think, before I felt like, it is justified and a very good title. So that's for readers who wonder what might be going on. All becomes clear as you get through, and there are so many topics. And as Joanna said, I know we're going to be talking about molecular phylogenetics, endosymbiosis. I love learning all these new words. Horizontal gene transfer. Let me see what else. Evolutionary trees, changing views on biology and evolution. And I couldn't decide where I wanted to start, so I decided I would start with a big essay question, a sort of philosophy of science that kept coming back in my head while I was reading about all of these ideas as you introduced them. The subject is an aspect of the scientific perspective or the scientific process that I don't think the general public always realizes is at work. And the proposition is that in science, a productive but incorrect idea or theory can be more valuable to science than a correct but sterile idea.
Absolutely,
yeah. Open your blue book and illustrate using examples from your book. And since you've already mentioned Carl Woese, he's the one that leaps to mind. So maybe we should talk about him.
Right in the middle of the book and of the subject that you raised, Jeff, is the tree.
Yeah.
So there is this ancient phrase, this ancient image idea called the tree of life. It goes back to the book of Revelation. It's been used in medieval religious art, the tree of life. And in its religious application, in the book of Revelation, it seems to apply to Christ, and the fruit and the leaves of the tree of life are the gifts that he brought to humanity. It's kind of an opaque metaphor, but that seems to be what it suggests. And then through the Middle Ages, people talked about the tree of life, and then you heard people talking about family trees. The history of your family could be presented as a tree. And in the Renaissance and early modern period, 17th, 18th century, a bit after the Renaissance, you had naturalists who were starting to try and classify creatures on Earth. Carl Linnaeus, the great classifier, and others were saying, okay, we've got a lot of different plants, animals, and other kinds of critter on this planet. How do we put them into order? How do we organize our knowledge about them? How do we put them into boxes or alphabetical order or some system where we can retrieve the knowledge that we have about them? One of the answers to that question was, well, let's put them on a tree, and all of the things that are similar by way of being a certain kind of flower we'll put over on this branch with these twigs coming off of it, and over here we'll put animals that have 4 legs, and we'll call them quadrumana, and we'll put them all together. So it was a system of organizing knowledge, but there was no evolutionary component to it. There was no sense that these things had changed over time and arrived at their differences and their similarities, until a little hint of that in Jean-Baptiste Lamarck, the early evolutionary theorist who had ideas about evolution, most of which were proven either wrong or too vague to be useful. And then after him, Charles Darwin, who was the creator of the first really modern evolutionary trees of life. He drew a simple sketch in his 1837 notebook of a sort of a stick tree, and he labeled the branches A, B, C, D, and he wrote above it, I think. And what he was saying in that notebook is, I think that species have evolved, they have diverged from common origins, diversified, and they're related to one another on these lineages that have changed over time, like limbs or branches on a tree. And then in his when he finally published his book, The Origin of Species, there is a tree in that, and there's an image that he offers, and he also says this. He says, the affinities of all the beings of the same class, these groupings, have sometimes been represented by a great tree. I believe this simile largely speaks the truth. That's Darwin, 1859. So that is a scientific idea. Some people call it a hypothesis. Some people call it a model, an illustration of Darwinian evolution. And it was very, very valuable at its time and for 130, 40 years afterwards. Just as Isaac Newton's principles of physical dynamics and movement of bodies on Earth were very, very valuable and were correct to the extent of measurable evidence until Einstein came along with relativity theory and he modified Newtonian mechanics. Just as that happened, Carl Woese and the people who were influenced by him came along in the late 20th century, starting around 1977, and they challenged and modified and corrected the tree of life, the Darwinian tree of life, so that now we understand, and this is a central subject of my book, that the history of life has not simply been like a tree with one common origin, the trunk, and then major limbs, and then branches and smaller branches and twigs, always diverging. There have, in fact, been limbs growing together, limbs growing sideways, branches growing sideways from one major limb into another, genes moving sideways across boundaries from one species to another, from one kingdom to another, and not just vertically from parents to offspring. And we can talk about that. You already mentioned horizontal gene transfer, so that's what I'm alluding to.
Yeah, we'll get
so that's my blue book offering to your question. The tree of life, as emended and corrected by Carl Woese and the people who have followed him, is a classic example of how science is a work in progress, and every idea, no matter how firmly it is established with evidence, is always still subject to challenge when new evidence comes along.
Now, when you have such a fertile and powerful idea, which leads to all of these new experiments, new ideas, lots of productivity, it's almost invariably going to be true, isn't it, that that idea can also get in the way of progress at a certain point because we're so attached to that idea now and is so firmly entrenched that modifying it might seem like heresy and giving it up seems like revolution, that we don't want to go there.
Absolutely. And that's what scientific revolutions have to contend with, a great deal of inertia. Once a good idea gets firmly established with a lot of evidence and with generations of scientists who have grown up on it, who have learned it, who have embraced it, who have based their lives' work on it. That's as it should be. Science shouldn't be having a new revolution every year, every week. Things should, once they're established, they should remain established until challenged by really serious substantive counter evidence. And another example of that, I mentioned Carl Woese, this important microbiologist at the University of Illinois. He, before horizontal gene transfer was appreciated, before the tree of life as a general shape was challenged, he came along in 1977 and challenged another scientific orthodoxy. It had been thought until then that there were two major kingdoms, two kingdoms that encompassed all life on Earth, essentially the bacteria and everything else. And bacteria were simple cells with no really complex internal structure, and everything else was composed of complex cells that had a cell nucleus containing the DNA and that had other internal organs that allowed this cell to function in complex and highly energized ways. That branch, the fancy name is Eukarya, or the eukaryotes, E-U-K-A-R, meaning pure kernel, true kernel from the Greek, true kernel referring to the fact that they have a cell nucleus. Anyway, two major kingdoms, bacteria and eukarya, bacteria and everything else. Carl Woese makes a discovery in 1977 and announces to the world through a scientific paper that no, there's a third kingdom. There's another group of animals, or another group of creatures, not animals, that we have overlooked. We have mistaken them for bacteria forever. Through a microscope, they look like bacteria. But with my new primitive gene sequencing methodology, looking at their genomes, I have established that they are not only completely different from bacteria, but they're more similar to everything else, the eukaryotes, complex creatures, than they are to bacteria. And that group of organisms eventually became known as the Archaea. Archaea as an archaeology, reflecting the fact that people thought maybe they were the oldest form of life on Earth. So that was the first big revolution triggered by Carl Woese. Not 2 kingdoms of life, but 3 kingdoms, including this group Archaea that had been completely unrecognized before 1977.
And his tree that he still drew had, what, 3 big trunks that sort of
3 big limbs, I would say.
Limbs.
One trunk and 3 big limbs. Yes.
Yeah. Now, and his method was pretty primitive, but virtuosic.
Oh yeah.
It's pretty amazing to read the description of it.
Is it?
Any idea of gene sequencing had come along. People forget those days if they're too young.
Right. People, yeah. People you hear this phrase gene sequencing now, and what it refers to is fancy machines in laboratories, and an undergraduate, a smart undergraduate biology student, can go into a laboratory now and using these automated gene sequencers can take a sample and sequence the genome of an organism. What, Joanne, in an afternoon?
Yeah, for sure. These days,
and for not that much money.
Yeah.
Right. But that's new. That began in the 1990s. In the 1970s, when Carl Woese was working, they didn't have those machines. So he had this deep curiosity. He wanted to understand the early history of life on Earth. And he decided the way to do that was by sequencing genomes of different organisms, or at least parts, fragments, samples of their genomes, and then comparing them, one organism to another. So he worked up this system based on earlier work by a wonderful British biologist named Fred Sanger, the great Fred Sanger. And Carl Woese adopted this method from Fred Sanger, and it involved toxic chemicals and explosive solvents and high voltage and radioactive phosphorus, and all of this stuff is going on in Urbana on the the 3rd floor of Morrow Hall, right, Joanne?
Yep.
Yeah. OSHA didn't exist at that point, or OSHA would have had a nightmare about this. And using this methodology,
radioactive stuff.
Yeah, right. And some of these people who worked with him, the men and women who helped him, are still saying, God, I'm lucky to be alive. The crazy things that we were doing. We'd get these shipments of radioactive phosphorus every other Monday, and we'd be pouring them into these electrophoresis units and using high voltage to separate fragments of long molecules, fragments of DNA and RNA, fragments of the information molecules, and then using them to compare one organism with another.
Now, so this is the early onset of what you came to be described, or you could describe as the molecular phylogenetics, which is the idea that you can look at these molecules, in this case the RNA, and
RNA,
and that looking at those can help you establish evolutionary connections and lineages. Is that about right?
Yes, that is right. I mean, sometimes I joke about this book and say, everybody wants to read a 400-page history of molecular phylogenetics. But this is the lighter side of molecular phylogenetics. Well, people, don't turn off your computers. Don't go away. That's just a fancy way of saying that Carl Woese and a few other people, including the great Francis Crick, and including Linus Pauling, that maybe you've heard of, they decided in the late 1950s and 1960s, how can we learn about the shape of life's history? What's related to what? Where the great limbs and branches on the tree of life are. Let's do that using the information reflected in long molecules, because long molecules are replicated but they're replicated always with a few mistakes, so you can trace the changes in these crucial long molecules, long proteins, DNA, RNA. And Francis Crick called it protein taxonomy because he was thinking about proteins and using taxonomy as a classification of creatures. Francis Crick suggested that maybe we should use the structure of proteins rather than, you know, the color of wings on a butterfly or the number of hairs on the nose of an ant. Maybe we should use proteins inside them as a way of sorting out species and seeing who's related to whom. Woese picked that up. Crick was a friend of his. And he said, I'm going to do that. I'm going to do phylogenetics, building trees of life, but I'm going to do it using long molecules. And one of the most important things Carl Woese ever did, a brilliant insight, was to pick one particular molecule and make that his Rosetta Stone molecule. A molecule so basic, so deep in all forms of life that it has changed relatively little over the 3.5 billion years of life, cellular life on Earth. And this single molecule, there are different versions of it, slightly different versions in bacteria versus in, you know, complex creatures, but it's essentially the same molecule, and it's a molecule of RNA, not DNA, meaning it's a single-stranded molecule, and it is part of the structure that all creatures use to turn DNA information into body parts, into physical life, into proteins. It's part of the translating mechanism.
So it made sense to use it because it would be, it's such an ancient molecule that it appears in virtually anything you want to.
It's conserved, as we would say. Yeah,
it's conserved, right? Yes. So it has changed over time, but it hasn't changed very quickly. Therefore, you could use it to go down and look at the deep branching, the deep limbs off of the original trunk of the tree of life. And so Woese brilliantly hit upon the idea of using that particular molecule. Joanne, should I say its name?
Yes, go ahead.
Okay, here it is. This molecule is called 16S ribosomal RNA. 16S ribosomal RNA, or in complex creatures, it's 18S ribosomal RNA. That's not going to be on the quiz at the end of the hangout, so don't worry about it.
But it's all the biologists happy, though.
Oh, come on. There were some bacteria in there that had much harder names than that.
That's true.
Oh, yeah.
Much longer. But so Woese is doing this, and these amazing, what we call now, can look back and say was sequencing aspects at a time when we barely knew how DNA was working, what its role was, although the idea of genes had been around for a long time, which I always find very interesting. But he did find some similarities, and then what led him to this third kingdom was finding some differences that just didn't fit into that idea of the lineage, right?
Right. He would take this molecule. His assistants would extract this molecule from one form of life, say this kind of bacterium A, bacterium B, bacterium C, and then maybe a simple eukaryote like yeast, which is a fungus. So it's a complex creature, but it's still a microbe. And he would cause to be extracted from each of them this particular molecule. They'd purify it, they'd bust it into pieces using chemicals, and then they would put those pieces on an electric racetrack. And I don't know if anybody's heard of electrophoresis, but electrophoresis is an electromagnetic way. Is it fair to call it electromagnetic or just electric? Anyway,
you set up a strong electrical field.
You set up an electrical field and you pull on these molecules, and they have charges, these fragments.
Yeah.
Anyway, it's like the Kentucky Derby of amoeba. It's like you take 15 amoebae and you put them at a starting point at Churchill Downs, and you open the gates and you fire the gun, and then these amoebae come out and they race one another down this racetrack at different speeds. And at the stopping point, you can see which one made it how far.
At different speeds, depending on their constituent parts.
Different speeds, yes, essentially containing
right?
So it
Charges.
separates them.
depending on their charge, which depends on their size and other things.
Yeah.
And then you turn the racetrack sideways and you race them again. So then they move in a different dimension. They move apart. So then, and because they've been fed radioactive phosphorus, then you can lay an X-ray negative over the top of this racetrack, put it in a box for 24 hours, and you'll get a picture burned of these different little amoebae. And there are pictures of Carl Woese from that era. And there's one in my book staring at these big X-ray films up against the light box and looking at these different spots. And he's figuring out which spot has which fragments of the RNA code, and which fragmentary sequence of the basic 4 letters that compose all RNA molecules. There it is. Thank you. There it is.
Yeah.
That's what he's doing.
There we go.
So Carl Woese did this for hours and hours and hours, for weeks, months, for a couple of years. And he said, I think I've destroyed, occasionally at one point he said, I would walk home occasionally and say to myself, Woese, you've destroyed your brain again today. Because this was so laborious to get bits of information. So he would identify fragments of 4, 5, 6, maybe as many as 20 letters. And for each organism, he'd have a number of those fragments. Each of those was like an amoeba, but when you found out what the letters were, it was like a word. Yeah. And he would take those words and he put them together into a paragraph for one organism. Here's this paragraph. Another organism has a different paragraph. Some of the words are the same, some of the words are different. And he would compare them and establish how closely one was related to another.
But ruining his brain was how he got to his out-of-biology experience, ultimately.
Yes. And so he was doing that. He was looking at one of these films, just the way Joanne showed us, one day, and he thought he was looking at a bacterium with a long fancy name, a kind of bacterium that produces methane. There's a group of bacteria that metabolize carbon dioxide and produce methane as a waste product in oxygen-poor environments. Whole group was thought of as the methanogen bacteria. And he's looking at one of them, and he's looking at these different amoebae, these different fragments of its RNA. And he says, whoa, wait a minute. This is missing some of the basic, crucial words in a paragraph that every bacteria should have. And then he finds another of the basic things that's missing. And he says to himself, he says to his assistants, eventually he says to his collaborators, these things aren't even bacteria. There's something completely different. And that was the discovery of the Archaea, the third form of life on planet Earth, for which he made the front page of the New York Times, above the fold, on November 3rd, 1977, with his feet up on his desk, wearing a pair of Adidas. Famous photograph. It's also
I can't tell you how many times I passed by his lab in Morrill Hall as an undergraduate, and there is this old newspaper, cover just tacked to the bulletin board outside.
Really?
Yeah.
Now, I didn't know that until right now, that he had saved that. It was on his bulletin board, or somebody had saved it for him.
Yeah.
If I had known that would be in the book.
Yeah.
Oh, I should have taken a picture, but I didn't carry a camera around back in the 80s.
But that was, I call that his 15 minutes of, Andy Warhol said, in the future, everybody will be famous for 15 minutes.
Yeah.
Well, that was Carl Woese's 15 minutes of Warholian fame. He's on the front page of the New York Times. And it wasn't the high point of his life as a scientist, I would say. Maybe it was, but it wasn't the only high point. But it was the high point of his popular fame. And after that,
a little
went back to the laboratory.
That I think you may have skated over, which is that the announcement of the Archaea as a new kingdom of life came first through press release, followed by the paper. And I think I differed with you coming up as I did 30 years ago or so, in the science and things. Press releases, science by press release was extremely frowned on in scientific
Yes.
communities. And that's a general way of saying he would be ostracized. So I think that when he did it by press release, that would be a very, very big deal. And that would give people a lot of negative to overcome to get to the idea.
Well, and it did. And I think I say that in the book, don't I? I mean, his colleague, Ralph Wolf, who was also there at Illinois, a very eminent bacteriologist who is still with us, I understand, Joanne.
Mhm.
Yeah,
I think Ralph is
haven't heard
97 now.
Wow.
I hope he's still going to his office every day. And I interviewed him. He was very helpful to me. But he makes the point to me and others that this was a disastrous misstep by Woese to allow his funding source, which was NASA, the National Aeronautics and Space Administration, and the National Science Foundation, to issue this press release on the same day that the Proceedings of the National Academy of Sciences were publishing his paper. And people reacted just the way you're saying, Jeff.
Yeah.
There were a lot of people who said, this is bullshit. You don't announce am I allowed to say that on a Google Hangout?
Yeah.
Who knows?
Go ahead.
This is not the way you announce serious scientific results. Let the paper stand on its own. So many scientists were soured on this discovery because of the mode of announcement. And
But I
that
think
cost him.
You also described or gave an idea of what his motivations probably were that made perfect sense as well.
Yes. That story was another one that Ralph Wolf told me. Years earlier, Woese had made an important, he had created an important theory about the mode in which DNA is turned into proteins, a particular mechanism. And he'd been invited to a big fancy scientific meeting I think it was in France. And Francis Crick was there, and a lot of the other giants of early molecular biology, Francois Jacob and Jacques Monod and others were there. And Woese was little known at that point, and he presented this great idea on one day of the meeting right before lunch to these eminent people.
Mm-hmm.
And when he was finished talking, they all stood up and went to lunch.
Yeah.
And nobody asked him any questions. And it outraged and mortified him.
Mm-hmm.
And he told himself, and he also told Ralph Wolf, that's when I decided I would never allow myself to be ignored again. So that, according to Ralph anyway, according to Dr. Wolf, that was crucial in enticing him to allow the press release to be issued at the time of the discovery of the Archaea.
But I think it was about then that I probably made a note that I wrote down that said that one thing that we get from your book, another thing that we get from your book is the illustration that scientists, while they do science, nevertheless have human personalities.
Yes. Yeah, that's a crucial part of this. We've been talking about people and ambitions and motivations and friendships and competition and anger and resentment, all of that. And that's part of my message here. That's all part of science.
Yeah.
Science is a human activity. And it is like, as I say in the book, it's a human activity like, symphonic music and Major League Baseball and Grandmaster chess. And it involves a lot of very human motivations as well as evidence and hypothesis and falsification of hypotheses.
All those things.
And I wanted to make sure to bring that up. So I'm glad it's here, because not only did you mention, the more negative aspects of our human nature, but also the positive ones, the congeniality and, just having a chat over beer and how that helps ideas come about and going to each other's parties and
trust and loyalty.
Exactly.
And
all
sharing.
that is science.
All of that. Yeah. All of that is part of it. It's all part of science. Science happens.
And that you made it explicit. You said this happens in science.
Yeah. Well,
either way, positive, negative, you said this is what happens.
My mentor and exemplar on that is a great book by a great philosopher and historian of science. And the book is Science as a Process by David Hull. And it's a history of the founding of a new branch of science, one called cladistics, which is a particular kind of classification of creatures. using certain approaches to evolutionary evidence and certain aspects of logic. Anyway, it was very controversial when it first came in, and now it's pretty standard. Do you think that's fair to say? Yeah. So it is one. But in the beginning, when this was a challenge to other forms of classification, it was a bunch of young Turks fighting an uphill battle against the scientific establishment. And David Hall, in this book, Science as a Process, uses that as a case study for making the point that science, this is not an accident, this is not an anomaly. Science always works that way. It always involves personal attitudes, loyalties, resentments, as well as evidence and hypothesis testing.
Well, I was thinking, Joanne, do you think we're ready for endosymbiosis yet?
Yeah,
and certainly personalities come in there for establishing that.
I felt like you had three main characters in the book with lots of supporting actors, and Lynn Margulis was one.
She certainly is. She's probably the second most important character in the book, the great Lynn Margulis, who I never met Carl Woese, but I was lucky enough to meet Lynn and spend a little bit of time with her, as reflected in that photograph in the book, where she and I and a physicist named Eric Schneider and Edward O. Wilson are all on a larkish field trip through Yellowstone Park. And Ed is wearing the borrowed ranger cap.
Yes.
Goofing off. So Lynn Margulis is this wonderful scientific hero. There she is. There's Lynn, Ed Wilson,
there we go.
Eric Schneider,
Ed Wilson, everybody.
myself.
Yeah.
And we're standing in front of the Great Falls of the Yellowstone River. So Lynn, in 1967, she's a freshly minted PhD in microbiology, and she revives an idea that had been suggested earlier by some crazy and neglected earlier scientists, one American, one Russian, and a couple of others. I tell their history because they're just so wild and wacky. I had to put them in the book. But Lynn is the important character. 1967, she revives this theory with a paper that, according to her, 15 journals rejected before it was finally published. And this paper proposes that complex cells, the ones in those eukaryotic creatures I've been talking about, including all animals, all plants, all fungi, complex cells became complex by swallowing bacteria and turning those bacteria into internal organs, so that the bacteria became symbionts inside, permanent partners inside complex cells. Hence the term endosymbiosis, partner inside. And these swallowed bacteria, one lineage of them, according to this theory, turned into an organ. They call it an organelle, an internal organ, in complex cells called the mitochondrion, plural mitochondria. And it's a really important organelle in all complex cells. It packages energy for use in complex cells. And she said, look through a microscope and you can see these things are captured bacteria of some sort. So 2 billion years ago or something, one host cell swallowed a bacterium or was infected by a bacterium, and instead of digesting it or rejecting it, that bacterium stayed, replicated, and evolved into mitochondria. That's the theory as she proposed it. And then in plants, there are other kinds of these internal organelles. They're called chloroplasts, and they do the photosynthesizing in plants. She likewise said these are captured bacteria. 1967, she publishes a book in 1970. Some people read it and say, this is really interesting, this could be true. A lot of other people read it and say, she's nuts. This is not possible. This is crazy. This crazy woman now, by that time she was, I think, at Boston University, and then she eventually moved to University of Massachusetts in Amherst. Lynn Margulis is the founder, reviver of the theory of endosymbiosis, was a very vivid, forceful, and controversial figure for a long time. She had a lot of chutzpah. She was very confident. She wasn't scared to press her ideas. So she fought battles in the scientific literature, in the popular literature. She lectured, she debated, she'd do anything to argue her ideas. And yet she was a ton of fun. She was a wonderful, charming woman. And then when genome sequencing arose, and people started using Carl Woese's methodology to look at some other questions, a fellow named Ford Doolittle, an American who by then was tucked away in Halifax, Nova Scotia at Dalhousie University. He had known Lynn a little bit when he was at Harvard. He said, I think I'll use Woese's methodology to test Lynn's hypothesis, her theory. And so he looked at chloroplasts, and he looked at their genomes, and he said, by God, these are bacteria. Or they're descended from bacteria. They're captured bacteria, because the genome of a chloroplast is completely different from the nuclear genome of the plant that it's in, and yet it's a close match to the genomes of a particular group of bacteria that I can name, he said, and they are these cyanobacteria. These used to be called blue-green algae. Right. And so he confirmed, using a sort of a Woesian methodology, what Lynn Margulis had proposed using microbiology.
But you still have to, I think you do it, but do you find that you still have to help non-biologists like me get over the astounding fact that eukaryotic cells, human cells, for instance, have different parts in them, and the different parts have different DNA.
Yes, it's very counterintuitive. That was my reaction too. This is crazy. Wait a minute. I mean, I had heard about Lynn Margulis and endosymbiosis probably back in the 80s. I was writing a column for Outside Magazine on natural sciences, and I read some Lynn Margulis back then. Never became acquainted with her, but I thought that's pretty interesting.
But in the popular presses, people hear about genetic characteristics that are inherited, but only through the matrilineal line.
Yes.
Which is the same idea, but I'm not sure they make that connection.
Well, you hear people talk about mitochondrial Eve.
Yeah.
The last common mother of all humans on Earth. Why do we call her mitochondrial Eve? Because we have traced back to our understanding of her using the DNA that's in mitochondria, which is completely different from the DNA that's in our nuclei. Why is it different? Because it comes from bacteria. It comes from a captured bacteria. It's been evolving not completely separately. It has transferred some of its DNA to our nucleus and vice versa, probably over the hundreds of millions of years, several billion years. But it is distinct, and that's why they talk about and it goes down only through the female lineage. Why is that? Because mitochondria are abundant in eggs and non-existent or almost non-existent in sperm. So the mitochondria you get from your mother, and you get your mitochondrial DNA from your mother. She got it from her mother, who got it from your great-grandmother, who got it from a bacterium.
So at some point.
And so you mentioned Frank Doolittle as he's going by. Wasn't he the one that, from which we get the interesting idea that you mentioned, that important idea that basically creatures and DNA have separate existences. They evolve separately.
Well, yes. First of all, Ford Doolittle, not Frank Doolittle.
Sorry,
Ford Doolittle. I think Frank Doolittle was a right fielder for the Yankees. Ford Doolittle is this fellow in Halifax. And he's talked about what you just said, but I think what you just said, Jeff, is if I heard it right, is also something that probably goes back to Richard Dawkins and the selfish gene.
The idea, in a way, that genes have their own agenda, basically, but also that when we get to looking at the tree of life as we now understand it, it gets tangled because genes evolve on their own way, in their own ways, separate from the animals and the creatures that are involved with them.
Well, separate I use separate in a lot of ways in this book.
I shouldn't say separate. Independent.
They have their own interests.
Yes.
We don't want to we don't want to anthropomorphize DNA or RNA, but there is a lot of reason to believe Richard Dawkins' idea of selfish DNA, that that DNA replicates itself. Why? Because it wants to make more copies of itself. Wants.
Yeah.
Why? Because it wants evolutionary success.
Yeah.
You know, the winner is the one who survives and leaves the most offspring. Well, with DNA, it's the same. The winner is the one that survives and leaves the most offspring, leaves the most copies of itself. So, boy, that could get us into even deeper weeds, and we could start to talk about transposons.
Yeah, well,
you
we
probably should, because we
Yes.
want to make sure to get transposons and horizontal gene transfer.
But wait a minute, before we go into transposons, let's talk a little bit, just so we don't confuse people. Let's talk a little bit about horizontal gene transfer.
Yes.
'Cause I think that's partly what you had in mind when you asked that. Is that we now know that we humans and all other complex creatures are composites. We are mosaics of different genomes from different sources.
Yeah.
And the entirety of our human genome does not come down to us vertically through the lineage of animals. Some of it comes sideways into the lineage of animals or into the lineage of primates by horizontal gene transfer. Genes moving sideways and getting into our lineage. A gene can appear in the lineage that way, and it'll be in a creature, but it wasn't in the creature's parent or grandparent. That's horizontal gene transfer. How does it happen? And I first read about it in 2013, I think, Joanne, right after I probably was visiting with you about the first time about spillover. And I was looking around for my next book project, and I started reading about horizontal gene transfer. Maybe I saw something that Ed Yong had published in his blog, or I saw a scientific paper or something. And I said, wait a minute, wait, horizontal gene transfer? No, that's not possible. That was my reaction in 2013. And I had already written 3 books or so about evolution at that point. I hadn't heard of it. So I start reading into it. And I discover this character, Ford Doolittle, not the outfielder, but the molecular biologist. And I start reading his work, and it takes me a little deeper into horizontal gene transfer. And then I discover this character, Carl Woese, at the University of Illinois. And now I start to see this great web, and I see that he's the spider in the middle of the web. And I, in a reckless, foolhardy moment, I say, I think I'll write a book about this. But so horizontal gene transfer, how can this happen? Well, there's a hint in a phrase coined by a great biologist named Joshua Lederberg, and he called it infective heredity. Infective heredity. So that gives you a hint. There are several different mechanisms by which this can happen, horizontal gene transfer. But the general proposition is that it happens by infection. Infection possibly by bacteria, infection by viruses. Viruses, for instance, can essentially perform a sort of drag and drop function on DNA. If a virus infects one particular creature and gets into its it gets inside its cell and invades its nucleus, it can acquire a stretch of DNA from a creature, and then make it part of its own genome. Then when that viral particle replicates, the alien DNA is replicated, and if the descendants of that viral particle infect another creature and get inside its cells, they can potentially drop that DNA in that cell, and it becomes part of that creature then. And that's a particular one particular method of horizontal gene transfer that goes by the fancy name transduction. Transduction, carrying things across state lines and dropping them illicitly in the wrong genomes. So there are several different mechanisms like that. But if you think of infection getting into the genome, that gives you a little bit of a handle on horizontal gene transfer.
Which, in your tree picture, then complicates things a lot, because now we have genes going back and forth, and it was Robert Feldman I was thinking of. Each gene has its own history.
Yeah.
And my addition is, and it's not necessarily the same lineage as that of the animal you find it in.
Right. Absolutely. Absolutely. There is a gene, for instance, and I discuss this near the end of the book. There is a gene in the human genome that is responsible for creating a membrane between the placenta and the fetus in humans, in human females, that's entirely essential for successful pregnancy. This membrane between the placenta and the fetus helps to transfer nutrients in to the fetus and carry waste products out of the fetus so that the mother can, into the placenta, so the mother can get rid of those waste products. Also probably protects the fetus against the mother's immune system, because the fetus is only 50% similar to the mother's genome. The other 50% comes from the father, and the female's immune system would be inclined to attack that alien genome. So this membrane is there, serving these important functions. And this membrane, we now know, thanks to the work of a wonderful scientist named Thierry Heidemann on the south side of Paris, and his group, his colleagues and students, we now know that that gene comes from a retrovirus, and that it was captured in human in primate genomes, in early mammal genomes, several different versions of it. It's been improved upon and replaced, but essentially it's been acquired from a retrovirus that infected mammal genomes and came to stay and made this possible. And in the viruses, this particular gene is an envelope gene, meaning that it creates a membrane surrounding the viral capsule. It gets captured in the mammal lineage or the pre-mammal lineage, and that creating that envelope is transmogrified into the creation of this membrane between the placenta and the fetus. So human pregnancy is not possible without this gene that we got sideways from a retrovirus.
Which is
right,
certainly
from the envelope gene. Yeah, it's pretty
startling development. Do you think you're ready now to spend the last 4 minutes summarizing everything about what is a species and what is a human being?
Is that all?
Yeah.
Well, I was more interested in actually sharing, like you wrap up the book by talking about Carl Woe's life. And I remember, and I'm just sharing this personally.
You knew him. You knew
that
him.
I knew him.
Yeah,
And
tell us.
well, as an undergraduate, I remember meeting him, and he was always, willing to talk to students. So, and he just said, I was working in somebody's lab, making auger plates, doing, things that they would put for undergraduates. And I was sitting at the autoclave, and he just comes up and we started talking, and he said, did you ever consider maybe life is not becoming more complex, but it's becoming less complex? They would just say this interesting stuff. So, and it did make me stop. I didn't know what to say, because here's the great Carl Woese. We all knew who he was, right?
Yeah.
But the other thing that was interesting was, as I got into social media and science communication, sort of he figured that out. Who is this person? And he wanted to get me on board with his crusade to end the swindling of Alfred Russell Wallace. He really was on Wallace's side.
Yeah,
that Darwin had stolen, and he wanted me to help get this word out, and gave me a book, and he had this big, invited the author of the book, and
I remember you telling me that when we had barbecue in Urbana. So let me talk a little bit about that too. First of all, he was so nice to you.
Yes.
So outreaching. And he could be that way. And I never met him, but I've talked to a lot of people. As I say in the book, he was my Citizen Kane, and I was, I was the person who went around and said, "Who was this guy?" And some of our viewers will know the movie Citizen Kane and understand that. I won't bother to explain it, but he was this mysterious figure, and all the testimony I got was that he could be really sweet. He could be really generous. He could be a really great mentor, teacher, and he could also be a crank and aloof and reclusive. and difficult. He could be all those things. He was a complicated man.
Yeah, I heard all those things, but my personal experience was he was very kind.
Yeah. And so, so he's the mysterious fellow in the middle of this book who made some hugely important discoveries and made possible hugely important discoveries by other people who followed him, adopted some of his methods, challenged some of his ideas, worked against him, worked with him. He triggered this revolution in molecular phylogenetics. And as a result of that, we know now that the tree of life is a pretty good approximation for the history of life on Earth, but it's imperfect, just the way Isaac Newton was imperfect, and it has been importantly modified into what I call a tangled tree, a different picture of the history of life on Earth.
Yeah, it was a great book, and I always love, as I tell Jeff, like when I read a book on topics I know, I'm reading it to see how the story's told. And of course, you are a great storyteller, David, and you did a magnificent job on that.
Thank you.
So I'm usually not there to go, I need to learn this horizontal gene transfer. Well, I already knew about it, right? And not all the players, but yeah, so I loved it.
Thank you very much. Thank you.
You know, it works well for lay people like me, physical scientist who has heard of cells and things like that, but isn't always so clear on all these things. I got a lot out of it too, and it's very important stuff. And it assembled a lot of ideas very well, and also brought these themes that I was interested in about how science operates and how personalities make things happen, and now it's a human enterprise.
Well, thank you both.
Yeah, no, it's fantastic the way you put it all together. Is there anything you'd like to add before we sign off, David?
I guess we may have said this already, but there's a lot of technical stuff that we've talked about, but this is meant to be a story for everybody. This is meant to be something that makes you want to turn the pages to learn not just about these counterintuitive, boggling discoveries, but about these very human people, these men and women who were so devoted and so curious and worked so hard in such difficult ways to make the discoveries. It's a book about, as I say, the most important biologist of the 20th century that you've never heard of, but also a lot of other very, very important biologists that you may or may not have heard of, like Lynn Margulis and Barbara McClintock and Ford Doolittle. So it's a book of stories and personalities and narratives and science.
Science, yes. And I have, you described some of these things so well. They're like in my head now, including parties and things like this. They're like, I'm reading a science book, not expecting to learn about, parties they went to. So it's all in my head. It's very good. Very well done. So any idea what you want to work on next?
Well, I've got I haven't decided on the next book project for Simon & Schuster, who's my regular book publisher, but I've got a 5-story assignment for National Geographic on the topic of the last wild places on planet Earth. So I'm in the midst of doing those stories while I do some book interviews and things. I'm in the midst of doing those, and those will eventually be pulled together into a book from National Geographic also, The Last Wild Places. So I've got plenty to keep me busy.
Important, though, right? We need to know what's still left out there. We haven't invaded.
That's what I do with my other hat. When I'm not following scientists through laboratories, I follow scientists through jungles and forests and swamps.
Well, thank you for doing that. So everybody, thank you for joining us and for and for talking, David, for talking with us today about your book, The Tangled Tree. And that if I'm in town, when you make it in town in a few weeks, I will make sure to say hi. So that
would be great. Great.
So
I hope I get to see you.
U of I features strongly in this book, so they're very excited to have you come by.
I'm very eager to get back there and very glad to be invited back.
That's great. Well, thank you, everybody. Thank you, Jeff, as always. And
thank you all. Yes.
We'll see you next time on Read Science.
Bye-bye.